Good day, everyone. Today, our group will be presenting a comparison between two types of microscopes, transmission electron microscope or TEM, and a compound light microscope. Our discussion will be divided into five segments. First is the comparison of the resolution and magnification of each device. Second is the unique components or mechanisms involved in each device. Third is the difference between their sources and the path of radiation. Fourth is the model or diagram of the devices. And last is the comparison of their modes of application and sample images taken by each microscope. One method for getting high-resolution photographs of both biological and non-biological specimens is electron microscopy. They are microscopes that provide an image that has been enlarged by using an electron particle beam to view specimens. There are two basic kinds of electron microscopes, the transmission electron microscope and scanning electron microscope. Their difference lies in the method of formulating the image, as transmission electron microscopes employ the transmitted electrons or the electrons that are going through the specimen to create an image, while the scanning electron microscope detects the reflected or knocked off electrons. As stated earlier, we'll be focusing on transmission electron microscope for this video. So, how does the TEM compare to a standard compound light microscope? For the resolution and magnification of the microscopes, TEM is considered to have both a higher resolution and magnification with more than 50 times 10 to the 6 times magnification. The CLM, on the other hand, has varying values depending on the model being used, ranging from 40 times to 1000 times magnification. In terms of unique, special parts or materials of the microscope, the TEM has the following unique features present. Firstly, it has an electromagnetic and electrostatic lens, which specifically allows the magnification to be manipulated by controlling trajectory and shape of the electron beam via electricity. Next, it has zinc sulfate fluorescent screen, which is a necessary feature especially to visualize the diffraction patterns, specifically the variations in the collision and electron intensities in the screen, which produces the light and dark contrast image. Third, it has a cooling system. Because the instrument employs high electric current, the cooling system is necessary to avoid overheating. For the CLM, on the other hand, it has a glass lens, which instead of employing electricity such as in TEM, magnification is manipulated by mechanically changing the objective. CLM has an ocular lens, whereas if TEM uses fluorescent screen to view the image, the CLM uses ocular lens to view the image. In terms of the light source and path of light, TEM uses an electron gun, usually containing a tungsten filament, which releases an accelerated beam of electrons. The transmitted electrons are then used to form the high-resolution image of the specimen. On the contrary, CLM uses incandescent tungsten halogen light bulbs, which produce light in the visible spectrum to illuminate the sample, which allows it to be seen using the lenses. Electrons from the electron gun travel through a vacuum in the column of the microscope. The electrons are focused by multiple condensers and pass through the sample, which then impact a detector. The path of light for CLM can be traced starting with the light source, whose light is focused by the condenser towards the sample or specimen's light. Eventually, this same light then is received by the objective lens, which allows the image of the sample to be magnified. Now that we have discussed the differences between the two microscopes, we proceed to their applications and sample photos of samples under both types. First off, light microscopes are very useful in the fields of biology, particularly microbiology. In general, light microscopes are useful when there is a need to visually observe the surface details of an object, or when dealing with living samples. Emphasis on living, more on that later. TEM is particularly useful in examining the inner structure of the samples. Detailed cellular structures and single molecules or atoms. TEM is particularly useful in material science and is particularly useful when dealing with non-living samples. So, why can we deal with living samples with the light microscope and not with TEM? One word: radiation. Recall that the light microscope uses visible light as its mode of radiation, whereas TEM uses beams of accelerated electrons. You can use TEM to investigate living samples, but whatever is left after is definitely not living anymore, which could skew the results. Proceeding to the pictures, we have the first set as follows. These pictures are microscopic presentations of the vegetative cells of Symbiochlorus hyanensis, a unicellular algae. On the left, we can see the pyrenoid P, nucleus N, chloroplast CH, and cell wall CW of an algae cell, as seen under a light microscope. On the right, we can see the aforementioned organelles with the addition of the vacuole V, as seen under the 10. From image D to F, notice how the organelles appear more defined and detailed as the scale decreases from 1 micron to half a micron to 100 nanometers. One cannot see such detail with a light microscope, but a TEM makes it possible. Remarkable, is it not? But CLM isn't necessarily less useful than TEM, the same way that TEM does not automatically mean better than CLM. The keywords here are practical and appropriate. Each microscope is designed for specific tasks, and it all lies to the researcher themselves to know when and how to use them. Like in the real world, the secret to taking good pictures does not lie within your phone alone, but within your subject and your skills as a photographer. So, as a microbiologist, one has to master the techniques and skills related to the sample preparation and to maneuvering the instrument itself when it is necessary and appropriate for the experiment.